Hello everyone, myself Vishampati Joshi, and today we are going to study about one of the important topic of the chapter, excretory product and their elimination. In this chapter guys, there are many parts, but the most important and one of the most conceptual part is counter current mechanism that we are going to study today, and that topic comes under the name of concentration of concentration of filtrate. That topics come under the name of concentration of filtrate. So as we have already studied, nephron are the parts where the urine formation occurs, and the concentration of filtrate, that is the concentration of urine, must also occur in the nephron. Now as we know, nephron have different structures, that is glomerulus, PCT, loop of Henle, DCT, collecting duct. Okay, but the most important thing that we will study right now, we will focus right now, is that of how to concentrate the urine, why do we need it, and how that occurs. So when our body, as we all know, our body requires water. Now when our body requires water for every day to day activity, for everything we require water, right? But when we dehydrate or when we stop drinking water, there is scarcity of water in our body. Now to prevent their scarcity or to conserve more water, we require a mechanism known as counter current mechanism, which is very important to concentrate the filtrate and to save more water when our body is dehydrating. So on page number 295 of NCERT, under the concept of concentration of filtrate, we have the first sentence that NCERT says is that humans have the ability to concentrate urine. Okay, that means they have the ability, means they can concentrate when they need. Okay, need means when they are dehydrating, so when there is dehydration in the body, then we require to concentrate the filtrate, or then we require to concentrate the urine. Now this concentration of urine can be done by a important mechanism that we will try to understand now, that is known as counter current mechanism. Now what do we mean by counter current mechanism? When we are talking about counter current mechanism, that means suppose there are two pipes, this is one pipe, this is one pipe, and in both the pipe the water is flowing. Okay, and if the direction of water in both the pipe, or the flow of direction of water in both the pipe is same, then we call it as co-current. But if the direction is opposite, suppose the direction in this pipe is from down to up and the direction of water in this pipe is from up to down, then we call it as counter current mechanism, then we call it as counter current. And in our body or in our nephron, there are two places where counter current mechanism occurs. There are two places. Now what are these? What are they? Number one is between the limbs of loop of Henle. First one is between the limbs of loop of Henle. We all know that loop of Henle is made up of two limbs. One is descending limb of loop of Henle and another one is ascending limb of loop of Henle. Then the next one where the counter current mechanism occurs is between the limbs of vasa recta, between the limbs of vasa recta. okay, between the rims of vasa recta. Now, please try to understand carefully. A co-current or counter-current mechanism will only work when they are in close proximity. Okay, so two things. One, the counter current between these limbs, and the second one, the proximity between these limbs are very important when we are talking about the counter current mechanism. Now, in maintaining this counter current mechanism, in the previous class you might have studied there are two types of nephron. One is cortical nephron, another is juxtamedullary nephron. So, the nephron which helps in maintaining the counter current mechanism in this part is known as juxtamedullary nephron. Juxtamedullary nephron. Now, to try just to understand the juxtamedullary nephron, in the last class we have studied, for example, if this is the cortex part of the kidney, if this is the cortex part of the kidney, okay, then the juxtamedullary nephron has three parts. The important part is glomerulus, followed by PCT, then a long loop of Henle, then DCT, and then there is presence of a collecting duct. Then there is presence of a collecting duct. Understood? Now, to understand this in a more better way and to understand the most important topic of counter current mechanism, let's try to get deep into the mechanism and structure of this juxtamedullary nephron. So, basically we all know that kidney is divided into two parts. The outer part is known as cortex and the inner part is known as medulla. So, the outer part is known as cortex and the inner part is known as medulla. To understand this in a more better way, okay, let's just try to understand again. I divided the part into two ways as I had given there, okay, that is the outer part, that is the first one is outer part, which I call it as cortex part, and the inner part which I call it as medullary part. Okay. Now, the nephron, as we know, structure starts with a afferent arteriole which diameter is of course more than that which is going outside, and it is known as efferent arteriole, and this duct of capillaries which are formed is known as glomerulus. This duct of capillaries which are formed is known as glomerulus. Now, after glomerulus there is presence of the main filtering membrane as we have studied, that is the presence of Bowman's capsule, Yes, the presence of Bowman's capsule. Let's draw it again to be very perfect. Okay. So, there is presence of first an afferent arteriole, okay, whose diameter is bigger than that of a efferent arteriole. So, afferent arteriole bring the blood, then there is duct of capillaries, okay, then there is presence of another capsule known as Bowman's capsule, then there is presence of another capsule known as Bowman's capsule which actually filters the blood, followed by PCT, and then followed by loop of Henle, okay, which consists of, one is the descending lip, then it consists of one which is known as ascending limb, but remember, as we studied, the loop of Henle has again, they have two parts, one is the thick part and another is the thin part. One is the thick part, this one, and another is the thin part, and the thick part of ascending limb of loop of Henle is little bit lengthy than that of descending limb of loop of Henle, followed by DCT, which opens into a collecting duct, Understood? Now there is a term known as osmolarity. You must have studied that. Osmolarity means the concentration that is present in our body, and that is measured in milli-osmol per liter. Now when we are talking about cortex, bachcho, the cortex has a concentration nearly about 300 milliosmol, nearly about 300 milliosmol per liter. In the same way, the medulla also has about 300 milliosmol per liter. Okay, same way the inner medulla. Okay, as we are going inside medulla, as we know, as we have studied in the last structure, that we are going inside medulla. That means here we have 300, then again we have 300. As we are going inside, the concentration is same, because our body concentration is same. Now when there is less or dehydration of water, when our body suffers from dehydration of water, then what will happen? Then there will be secretion of a special hormone. That is known as ADH. So during dehydration, what did I say? During dehydration, okay, there will be secretion of ADH. What is the full form of ADH? Antidiuretic hormone. So what they will do? They will increase or they will ask the release or reabsorption. They will ask the reabsorption of NaCl from where? From ascending limb of loop of Henle, and this will happen actively. So if you remember, guys, this ascending limb of loop of Henle, what did I say? This ascending limb of loop of Henle, they have specific part which are permeable for electrolytes, So after ADH come, when there is dehydration in the body, ADH comes. After that, there will be release of NaCl. What? NaCl will come out. What will happen? NaCl will come out from the thick membrane. Remember that. So this secretion or this active secretion, this will happen from the thick membrane, thick part of ascending limb of loop of Henle. Understood? Now if that happens, what will happen? Slowly, as we know, now NaCl came out in the interstitium. When NaCl will come out, then of course the concentration will change. As we go down, more NaCl will get released. As we go down, more NaCl will get reabsorbed. Then this concentration will change to here 600, 900, 1200. So isn't a concentration gradient is formed as we go downside, the interstitium of the inner medulla, then there will be change in the concentration, and the concentration will increase. So the concentration will increase in both sides first there. So when the filtrate is coming, concentration is increased, and we all know that the descending limb of loop of Henle is highly permeable for water. So as concentration increase, and water always travels from lower concentration to higher concentration. Understood? That is lower osmolarity to higher osmolarity. So water will travel from lower osmolarity, that is from here to here. Understood? Okay, clear? Now once that happens, again the osmolarity is maintained. Now going back into the blood, we all know that the ultrafiltration which occurs here is non-selective. That means if we are considering the osmolarity of the blood, then it will be 300. As we know, the blood osmolarity or the interstitium osmolarity is always the same. So whenever it will come, it will be 300 here. Okay, now but if we are going downside, okay, then it will try to match the outer one. So it will be 300 here. Okay, it will be 600 here, it will be 900 here, it will be 1200 here. Okay, in the same way here it will again. Now the water came out, that's why it was concentration was increasing, but here the solute is also going out, that means solvent is also going out, solute is also going out. That means again the osmolarity or the concentration will decrease. So that means again here it will form 900, again here it will form 600, and in this way the osmolarity of inside will match that of the osmolarity of outside. Now another thing, if this is non-selective, then of course the urea will also come here. So let's just take, this is our urea, he is very happy that he is now inside the nephron. But when, as you have studied in reabsorption or selective reabsorption, 70 to 80% of all the nutrients, all the water are reabsorbed. Now here urea is now very unhappy that all friends, all bicarbonate ions, all water are reabsorbed. So he asked PCT, can I get out here? PCT said no, you are not allowed to get out. So he again came here. Now again he saw, again he is very unhappy that water went out. Again he came here, he asked this part, can I go out? No, he said no, you are not allowed to go. Then again he came here, okay, very unhappy. He asked, can I go out? This part said, see, I can allow you to come in. Yes, I can allow you to come in, but I will not allow you to go out. Then again he went to this part, everyone denied him, no, we will not allow you to go out. Then he reached this part, and then he became happy. He said, the lower part of collecting duct, what I said, the lower part of collecting duct is permeable for the reabsorption of urea. Now if you go and study the first page of this chapter, in the last paragraph, second last line, it is written that the urea, some amount of urea is reabsorbed to maintain the osmolarity of the inner interstitium. So going back, this urea, it became very happy, and the urea came out dancing. Urea came out dancing. Now try to understand, okay, if the urea will come out, then the concentration of or the osmolarity of the interstitium and the osmolarity of this urea will become same here. Osmolarity will become same, but here we know that the osmolarity is less. Why? Because NaCl is still present here, but after some time NaCl is removed, means the friends are removed. That means concentration of urea will increase as we go further, and here there will be higher concentration of urea, but here there is low concentration of urea. And this part said that yes, I can allow you to come inside. So urea, what will happen, it will again go back into this side, then again this cycle will repeat, then again this cycle will repeat. This is known as a common nephron urea cycle. Now this urea cycle will repeat to maintain the osmolarity. So now I told you that there are two things which maintain the osmolarity. Number one thing which maintain the osmolarity, number one ingredient which maintain the osmolarity is, okay, osmolarity is maintained by number one NaCl and number two, that is urea. Okay, so the concentration of NaCl and urea, they both maintain the osmolarity and they maintain the gradient or osmolarity gradient, that is important for counter current mechanism. Understood? If we are clear with this, okay, now please try to understand here what is the osmolarity, 1200, and urea is more. That means NaCl is less. See, to understand this, if I draw a table, okay, so what are the two things that are important? One is urea, another is NaCl. One is urea, another is NaCl. Now, if in the interstitium, try to understand, if in the interstitium urea is more, then NaCl will be less, because the total osmolarity they have to maintain is 1200. This is inside the limb of loop of Henle. Here urea is more, that's why it will travel inside. Here NaCl is less, that's why the NaCl from inside will travel here. That means in the thin segment, what did I say, bachcho? In the thin segment, the NaCl will come out passively. It will come out passively. So when I am explaining it here, the thick segment or thin segment of loop of Henle will allow release of or reabsorption of NaCl passively. Now question may be asked, okay, where it is released or reabsorbed actively? And the answer is from the thick segment, or from the thick part of ascending limb of loop of Henle. But when the question is asked when it is released in passive way, then the answer is thin segment of limb of loop of Henle. Did you understand? Now a very important work is done. All the osmolarity is maintained, but he is doing a lot of work, right? Now if I ask you to do a lot of work, you will start demanding, sir, I need some food, sir, I need some pizza, sir, I need many types of food. But in this way, nephron also demands food. So food will be brought by, of course, yes, blood vessels. So if for example we draw a blood vessel something like this, okay, let's take the red color and draw blood vessel something like this, okay, just to provide them food. Then what will happen? What is the osmolarity of blood? 300. So if the osmolarity of this blood is 300, then what will happen? We all know that the capillary walls, what did I say? The capillary walls, they are openly permeable. That means they allow very easy diffusion. So what will happen? All the NaCl which are released here, they will go back into the blood vessel. They will go into the blood vessel, and this will decrease, they will destroy this osmolarity which is present there. So what we can do, bachcho? Can anyone tell me how we can solve this problem? How we can give the food? How we can solve their problem by giving the food? So what will happen? What we can do, instead of drawing a single line here, can't we have a blood vessel which passes from here to here like this? Can't we have that? So when we are talking about that, let's go back into the blood vessel, okay, and I can draw a blood vessel something like this, which comes from here and goes back to here, which comes from here and goes back to here, something like this, okay. And this blood vessel is known as vasa recta. This blood vessel is known as vasa recta, okay. So this is the descending limb, am I correct? No. Whenever you are going to draw a vasa recta, its direction is always opposite to the direction of the flow of filtrate. That means if the filtrate is flowing this way, then the vasa recta has to be drawn from the side of DCT. Understood? Now what will happen? We all know that the vasa recta, what is their osmolarity? 300. So all the NaCl which was released from this place is again went back into the vasa recta. So here the osmolarity of the blood, of the blood in the interstitium of inner medulla, first it will be 300, of course, when it came. Then as NaCl will come, it will increase 600, then again it will increase 900, then again it will increase 1200. But when it comes back into the ascending part, that is near to the descending limb of Henle, this ascending part of vasa recta, what they will do, they will release back, they will release back the NaCl, in this way. Here they took, here they release. So don't you think this osmolarity gradient is maintained very well? So this is the way where a counter current mechanism is done. So what is their benefit? Nobody asks me. The benefit is that now let's go to the important part, or the important benefit bath. So when I am talking you about this collecting duct, then we all know that collecting duct helps in high reabsorption of water. Now try to understand, when the filtrate came here, the osmolarity is increasing. That means water will come out, and the filtrate which was present here, which was 300, which was 600, which was 900, and now it is 1200. So there will be increased, after release of water to the interstitium, there will be increased concentration of urine. So can I say as the concentration of filtrate, okay, increases, okay, due to high reabsorption of water from collecting duct, okay, and all this happens due to maintenance of a concentration gradient. Concentration gradient. Okay, so initially when the filtrate was how much concentrated? 300. So if I take initial concentration is 300. Okay, final concentration is okay, after this counter current mechanism 1200, right? So if I ask you how many times we can increase the concentration of filtrate or urine, then it will be 1200 by 300, that way 4 times. Okay, so a human body or a nephron can increase 4 times concentration of the initial filtrate, or we can say the body or the nephron, the kidney can produce 4 times concentrated urine than the initial filtrate. And in this way, when the water is reabsorbed, we can save a lot amount of water. And this is the whole process of counter current mechanism, starting from here, the basic things, to the final part of diagram, that is the counter current mechanism, and this is the final product, that is 4 times concentration of urine. So I hope you understand this basic part of counter current